Welcome you all to the next class on Inorganic Chemistry of Life, Principles and Perspectives. In the past two classes I have been trying to explain the electron transfer in general as well as in electron transfer with reference to the enzymes. Okay, now having learned the transport storage of oxygen, transport storage of iron, transport of electrons, etc. Of course, you do not store electrons. So, with all these now we are very well equipped to go to the next topic, which is the enzyme reactions of iron, which are oxidative reactions, reductive reactions and hydrolytic kind of reactions. All these three we will see some example or the other. We will start with oxidative type for the first as a first part of it, and in the oxidative type these are referred as the oxygenases. So, oxygenases are the enzymes which oxidize substrate by using one or both of the oxygens of the O2. It will use either one O of the O2 or both the O's of the O2. If it uses one O, it is called monooxygenase. If it uses both the O's, it is called the dioxygenase. So, that means monooxidation and dioxidation, and that is what basically means. So, when it does the monooxygenase property, that means only one oxygen from the O2 is used for substrate oxidation, the second O will go as water. In case of dioxygenase, both the O's of the O2 are utilized for the substrate oxidation. Just look at an example, some kind of heterocyclic kind of a organic substrate, and this with oxygen and giving the hydroxylation of this, and the second oxygen will go as water. In fact, one can use this labeled one and find out the labeled water as well. So, this is called the monooxygenase because it is using only one O of the O2 to oxidize the substrate. Okay, second part of this is the dioxygenase. The dioxygenase means both the oxygens are added to the substrate. Now, in this there are two ways with respect to a double bond. This is the double bond. You can just add across the double bond in a equal way. So, or you can use outside this double bond. So, if you use within this, it is called the intradiol oxygenase, and if it is outside, it is called extradiol oxygenase. Okay. So, when the intradiol oxygenase, it will give this kind of a product, and if it is extradiol oxygenase, it gives the other product, which very well. So, we will look at later on some of these enzyme, their mechanistic aspects, etc. So, I hope from this slide you understand what is oxygenase, what is monooxygenase, what is dioxygenase. In the dioxygenase, you have intradiol dioxygenase, extradiol dioxygenase. So, how does that add? How does that add the O? So, how is O transferred? Or in other words, what is a O2 activation? See, why O is required? How can you get O out of the O2? So, O2 is a diatomic molecule with double bond. So, if we want to get from this O to minus, obviously you have to pump 4 electrons inside. So, if you do not have this, you will not get this. So, first 2 electrons will break one other bond, next 2 electrons will break the other bond, totally 4. So, that is because the number of bonding electrons minus n number of anti-bonding electrons, and that should become 0. So, in order to take the bonding out. So, when this happens, that will be the thing. So, in other words, the O2 add 4 electrons, and in presence of the 4 protons give 2 H2O. So, if there is no substrate, both the oxygen will go to the water. So, this is what we call as the O2 activation. So, as you can see, 4 electrons have to be added. So, you cannot add all the 4 electrons at one time. You have to add 1 electron, add 1 more electron, add 1 more electron, add 2 protons, add this. So, that will become 2H2. So, O2 1 electron will become O2 minus dot, 1 more electron becomes O2 2 minus dot, 1 more electron becomes O2 3 minus, because that will not be very stable, and that will break down to O minus and O2 minus, and O2 minus will combine with 2 hydrogen H plus and become H2O, and 1 more electron, that will become O minus will become O2 minus and take up the 2 protons and the water. So, as you can see, in case of this the reduction of the O2 or activation of the O2, you also have concomitantly protons are associated with it. So, all this depends on what? Depends on the redox potentials. So, the activation obviously depends on the redox potentials, and the redox potential in turn depends upon the in case of hemes, it depends on the axial coordination at the ion center. So, whether the coordination is more sigma donor, more acceptor, pi acceptor type, or a combination of these, that will determine how easy that you can get the electron, how easy you can reduce. So, that is most important, because the ion redox potentials will depend upon what is the surrounding, what is bonded immediately, what is the hydrophobic, hydrophilic environment that is being created. All these will influence the potential of that. So, therefore, so for example, if you take the coordination we have seen earlier, either you can have a thiol or you can have a histidine, etc., etc. So, thiol or histidine, when you compare the histidine versus the thiol or cysteine, histidine is a good sigma donor and pi acceptor. So, it controls the electron density flow to the ion center. So, therefore, the enzyme, the nature is smart and nature tries to arrange the requisite group so that the potential shifts as per the requirement. Now, whatever we have seen this this electron transfer process, let us look in terms of the potentials, then we will be able to discuss and debate. Now, you take O2, add 1 electron, it will become O2 minus dot, and what is the potential here? Minus 0.33, that means it is negative potential. Okay. So, if it is negative potential, what will happen? Delta G is in this is minus NF and minus 0.33. So, that will become plus 0.33 NF. So, this is what? Is this feasible? Is this non-feasible? Clearly, it is a non-feasible reaction. So, this means the first electron which we are talking about here cannot go easily spontaneously. But once, let us assume for some reason we are getting this in, how we will see, that is why the enzyme is required, otherwise you would have just thrown the electron into that. The next part is O2 minus, 1 more electron O2 2 minus, in presence of 2 protons it will become H2O2, and this is a plus 0.87. So, it is very plus large, therefore, delta G is minus, therefore, it is favorable. The next one, going from here with 2 more electrons and 2 more protons, you will get 2 waters, not 1 water, 2 waters, is again positive. So, in entire step of this, the first step is the negative potential. Suppose it has to go to O2 directly to H2O2, is a positive. O2 minus to H2O, that is also positive. O2 to overall H2O is also positive. So, that means the conversion of O2 to water is a favorable reaction, though the first reaction is formation of superoxide is not a favorable one. So, this is thermodynamically unfavorable, therefore, the protein has to bring down the activation energy and make it accessible, and that is where the protein's role is coming into the picture. Now, you understand how the O2, so O2 breaking down to O2 minus requires 4 electrons, then to become water 4 more protons. So, O2 plus 4 electron plus 4 H plus is to H2O, and that is it. Now, having studied the O2 activation, how the OO breaks, that now we understand. That is how the enzyme breaks it. That is how the enzyme breaks it. Enzyme adds first electron, then second electron, then third, fourth electron. Once it goes to third electron, the reaction is very fast, it will go to the fourth electron without any realization of that. Okay, and there will be a proton pump parallelly and protons will be added. Therefore, at the enzyme center the O2 can be. So, at this stage if you have a substrate, it can add the one of the activated O or both the activated O's can be added to the substrate, and thereby it will turn out to be a monooxygenase or a dioxygenase. Let us look at very popular enzyme of example. So, a popular enzyme for this is Cytochrome P-450. Why is it popular? So, why is that so popular? See, so cytochrome P-450. So, before we come to the question of why it is popular, let us come to the word cytochrome. Has got two words, cyto and chrome. So, cyto is cell and chrome is color. So, that is why these are the cellular proteins. So, cyto and color, and the color will be absorption, that will be in the nanometer, and P450 cytochrome P450 is 450 is the nanometer absorption. So, that is how nomenclature in cytochromes will come, and you do not need. Now, let us look at why is this cytochrome P450 is so popular. So, cytochrome P450 once upon a time was considered as a life saver, but later on it was found it is not true. The reason, why the reason? Reason is cytochrome P450, as I told in the beginning of this course, is involved in oxidizing the apolar substrates, nonpolar substrates, organic substrates to adds the hydroxyl group or oxidizes and makes it more polar or soluble kind of thing. Now, that is where the fatty acids, fats that you eat can be digested, that is where I talk to you. Now, we can also learn they are involved not only in converting those oil stuffs, a variety of metabolic intermediates in the cell. They could be lipids, they could be steroids, they could be hormones. All of these can be hydroxylated by this enzyme. So, the main thing is it catalyzes the oxidation, and cytochrome is a family of enzymes, and but though we are talking about this particular enzyme here. So, the main reaction RH is taken as some kind of a hydrocarbon, oxidation to H plus 2 electron. When oxygen activation, what did we study? 4 H plus for 4 electrons. The reason is you have to convert that into both of them into water. Here you do not need to convert into water. So, you need only one O to convert into water, that is why you require 2 H plus and 2 electrons, and therefore this RH. So, it is basically inserting one of the O into the RH bond, that will become ROH. Inserting one O of the O2 into RH bond. Okay. So, these are cellular, it catalyzes a lot of reactions, particularly of toxic compounds also, drugs, metabolites as I said. So, therefore, this is not a lifesaver later on people found, and if you see the recent books they will not write this as a lifesaver, but if you see the very old version of biochemistry books, obviously you will see that it is a lifesaver. Okay. So, this can be in that, and a lot of imidazoles and triazole kind of a molecules, they will basically inhibit the activity of these, they act as inhibitors of this. Okay. So, we need to look at some details of this enzyme, how it functions, etc. This is a very highly prevalent enzyme in the entire biological systems, and it is based on the cellular based systems, and it is involved in the oxidative process, and you can have the, in this case, the monooxygenase. Having studied some basic aspects of cytochrome class and cytochrome P450, let us look at the electron transport. So, this is in this particular scheme, you have some part here, some part here, some part here. You have 3 portions we are talking about. Okay, this is the last part where the cytochrome P-450 is involved, and this is the last part where RH is converted to ROH, and this is coupled with the heme cytochrome P-450. This part is connected with the cytochrome P-450. But cytochrome P-450 on its own cannot do. It has to get electrons and taken from something, that is taken from something, that is taken from something. So, you see that. So, at the end, the beginner in this is the NADPH NAD plus. So, when NADPH oxidizes to NAD plus, what will do? It will release one electron. This electron will convert FAD, which is oxidized form, into FADH2, which is a reduced form. When this reduced form of flavin adenine dinucleotide turns back to oxidize, that will give one electron, and that electron will convert the flavin mononucleotide into flavin mononucleotide, which is reduced form. So, when this reduced form of the flavin mononucleotide return back to the oxidized form, that will transfer the electron. That will transfer the electron to cytochrome P-450, which is in the oxidized form, and takes up the electron, goes to the reduced form, and this in turn will break the O2 to one part of the O to H2O, other O is added to ROH. How it is done? That is called mechanism. I will be explaining in the later slides. Maybe after 1 or 2 slides later, I will be explaining how this O2 would give one O to this, one O to this. So, that is all. So, is that understandable? NADH, NAD plus is a standard source of electron, and this is this gives the electron to the FAD and makes the FAD reduced, and then FAD reduced when returned back to the oxidized form will give electron to the FMN, and then that is flavin mononucleotide will get reduced. When the reduced flavin mononucleotide returns back to the oxidized form, will give the electron to the oxidized form of the cytochrome P-450, and cytochrome P-450 will go into the reduced form, and that will give the electron, and then electron that will break the oxygen, and then you have one O going with this, other O going as the water. And this is the kind of electron. But generally we do not look at all these all the time, and we look at this. So, that means this enzyme must have a set of FAD cofactor, it should also have FMN cofactor, and then by the heme cofactor. Of course, NAD plus is always available in the cell, so that will give this one, but this cofactor and this cofactor has to be a part of that kind of things. So, why so many things? Why cannot this directly give to this? So, if you look at the redox potentials of this with respect to the heme, they will not match at all, they are highly negative, and so therefore, it needs to go through one more cycle, and then finally. As I told you in the previous class, the difference between the donor acceptor potentials within 0.2, 0.3 is good enough. Greater than that, it will not be a reversible electron transfer process. Okay, so is that clear? So, that is, you cannot have directly an electron going from NADPH to the heme because of the redox potential differences that we have. Now, let us get into this enzyme structure, other things, etc. Now, there are several cytochrome P-450 have been studied for the structure and the reactivity, but I will be taking only one example, that is cytochrome P-450, which oxidizes the camphor, So, therefore, this is the camphor, and this requires, as I mentioned, to NADH. Of course, the other equivalents are already there, H plus O2, and what is required? This enzyme cytochrome P-450 camphor, CAM refers to camphor, P-450 refers to cytochrome P-450 camphor. That will give a product where hydroxylation is here, not here, not there, not there, not there. It is hydroxylation is only here. So, that means it is the position selective, and also in this particular kind of a geometry. So, it is stereo selective and rigid selective. So, the product of this camphor upon catalysis by the cytochrome P-450, the product has a selectivity and specificity, and this is both regio and stereoselective. So, this is the total protein, and has somewhere here the heme, and then you have FAD, FMN, which we are not showing over there. Now, if you look at this, expand this region and look at, that is where you can see that. So, what is this has? This has the heme ion, there is other coordination here, and this side is empty, but very close proximity you have a lot of residues there. So, a lot of residues are there. Now, this is where what we need. How come this is getting such a very high selectivity in terms of regio and stereo? You see this particular thing. This is from a structure, not from a apoprotein. Before the substrate is bound, it is regular protein, but before substrate is bound. This is a structure after the substrate is bound. So, actually if you put substrate like molecule, you can always get the structure, because that will be inhibitor. So, you can see there are residues here and there, phenylalanine, tyrosine, all these kinds of phenylalanine, all these residues are there. And this is your substrate, and this substrate is not just left alone, there are some interactions between these ones and these ones. So, there are some hydrophobic as well as these interactions. So, all these interactions and the size of the core gives selectivity to recognize the camphor. If some other molecule comes here, it will not be recognized. So, therefore, that will not be considered as a substrate and will throw it out. So, the enzyme is active, it will understand what is its right substrate. How do it understand? From the weak interactions coming from all these, they will touch in some way, they will touch in the form of hydrogen bond, they will touch in the form of hydrophobic interaction, they will touch in the form of van der Waals interactions, etc. All these interactions will make it whether the right substrate has come or not. And this you can see that all of these are there. Because this is held like this, only this portion is accessible. Therefore, only that portion is exactly on the top of the ion, and therefore oxidation or hydroxylation takes place at here, not there, not there, not there, not any other place. So, how come is the camphor is getting the stereo and regio selectivity? The stereo and regio selectivity of the camphor is coming because camphor is held, in other words the substrate is held on this enzyme only in one fixed manner, where that particular carbon is straight exposed on top of the ion center. Why should it be on the top of the ion center? That will be answered when we get into the mechanism, because the ion center is the one which activates the O2, creates the active O species, therefore, it should be close to that proximity. So, therefore, this substrate and this position is exactly aligned on top of that. For aligning, this whole protein is playing a role. So, therefore, all these secondary interactions are absolutely important to this one. Okay. So, you understand that the selectivity, how it is coming. Now, let us look at a little bit more interior into this, that is the kind of a mechanism. How is this able to do that? So, as you can see that this particular is the starting step, and this is in the ion 3, and you have a cysteine on this side and you have a empty space on this side. But in the resting protein, the 6th position, which is empty, is never empty, is always with water. So, that is called, that will indicate it is a resting part of the enzyme. Now, this resting part of the enzyme, when the substrate RH is a substrate comes in proximity to that particular protein, in that the protein gets activated. So, protein starts thinking that I must do a reaction, and that is how it gets activated. So, it comes in the vicinity, you have already seen in the previous thing where it binds, how it binds, etc. So, now the protein is active, therefore, the steps start working. So, it will be ready to take up the electron from the electron transport series, which I showed you on the 2 slides behind. Okay. Once the electron comes, that will be iron 3 becomes iron 2, and this first step has been identified. How it is identified? This is trapped. How it is trapped? You just pass CO at this stage and then that will convert into one of the complex, and this complex has a absorption, IR, all kinds of things can be monitored and studied, and then that is studied. So, this is not a part of the, this part is not a part of the mechanism, this part is a part of identifying this intermediate. Now, once that becomes this ion 2, and now ion 2 is active towards the oxygen, because ion 2 can always shuttle to ion 3. So, that means it can add an electron. So, now this is ready to O2. So, therefore, now you see, tell me, suppose if this O2 is ready at this stage itself, what will happen before the substrate comes? Suppose at this stage itself O2 comes. Suppose if the O2 is a first step, what would you expect? If the O2 is first step, then the O2 gets reduced and that can add to the protein and protein can get destroyed. So, that is why it has to get activated only after the substrate comes. So, this is a substrate, then the activation, electron transfer, ion 2. Now, ion 2 now is ready, it will take up the oxygen and it will add one electron from itself into the O2. So, when you add first electron, that becomes O2 minus dot is superoxide. You know, this is favorable now because it is activated by the enzyme. This is activated by the enzyme, therefore, it is okay. It is not a free O2, it is activated by the enzyme. So, therefore, first step which is otherwise not favorable has been made favorable. Therefore, the role of the enzyme is to hold the oxygen, to activate the oxygen. Then at this stage one more electron will be added and that will become O22 minus. So, that is O22 minus. Now, the O22 minus already one bond is broken. So, it is very active in stage, can in presence of the two protons can take out this OO, break this OO into water and bring an internal redox. These electrons are not coming from the other internal redox. So, internal redox you require 2 electrons. So, therefore, ion. So, the ion gets oxidized either 4 plus and with the porphyrin radical or 5 plus. So, therefore, this thing is now activated, this becomes a ferryl oxo species. This oxo species is a very active species, it will very immediately add to the substrate and substrate becomes RH becomes ROH and then returns back. So, in all this from here the steps are very fast. So, this part and this part are very fast. So, the first part is the first electron and the O2. These are somewhat slower in step, very likely this may be the kind of a thing where you have determining kind of a step. So, you can see that all of these are the kind of intermediates and the spectrum overlapped over here for 1 and the 2, and that is how one can make out the difference, that is what is happening. So, what is happening? Now, this forms a ferryl oxo. Ferryl oxo will oxidize the RH to R dot and OH, then this will become ROH and this is kind of a rebound mechanism. Okay. So, you understand now ion is an ion through. First step should be the addition of the substrate should add. If the O2 gets added, then it is a devastation because it will get activated and the protein will get hydroxylated, that means protein commits suicide. Protein commits suicide. So, therefore, to avoid this, then first you have the substrate, then you have the electron transfer, then followed by the 2 electrons, then that breaks and then gives a ferryl oxo and then that continues to form this kind of a thing. Okay, so therefore, overall what you have seen that one is a ferryl oxo formation, other is the CH activation. So, up to ferryl oxo nothing will happen to the substrate is just being sitting there, but substrate has to be there in the beginning. So, now ferryl oxo is formed like this, CH can be that way, CH can be this way. So, okay, in either of this you can see in this kind of a thing you can have a transition state of this cyclic, and then this can break down into COH and ion. Or it can need not form a cycle kind of thing, it can form a linear bond of this, and this can break down to FeOH and C dot, and then this will recombine to form the COH, recombine to form. So, these things are possible. There are a lot of mechanistic approaches have already been worked out in the literature. It is not that the further details are not known, but for this particular course beyond this will be become a too much more. So, in this lecture what we have learned? We have learned there is a class of enzymes called cytochrome P. In that we have taken an example of cytochrome P-450. Cyto refers to the cell, chrome refers to the color, and here in this case the color absorption is at 450 nanometer, and these are all they are involved in oxidative process. They oxidize substrates, a lot of substrates, even metabolites, intermediates, drugs, all these kinds of things, and they give other kinds of formation of the product, and those are all dangerous products. In fact, I tell you at this stage I should bring one aspect to your notice. The cigarette smoke has the benzopyron. We know that the cigarette smoke is carcinogenic. As carcinogenic, people think it is because of the benzopyron. It is not true. It is the metabolite of the benzopyron where the double bond is converted to epoxide. That epoxide is the one which is carcinogenic, not the benzopyron itself. So, therefore, the P-450 cannot be lifesaver because it is converting a lot of drugs, intermediates, everything into OH, and therefore, that is the dangerous kind of a part. And then we have looked at the electron transfer. In the first substrate binds, then the electron transfer, then 2 electrons transfer peroxo. The peroxo will quickly break down to ferryl and water, and the ferryl will quickly add oxygen to the substrate. So, the slow steps in this are the substrate binding and the first electron transfer. All other processes are very fast and they go at a rapid rate. So, that gives and then I have talked to you the 2 possible ways of CH activation and the ferryl oxo species. The specificity and selectivity of the camphor is coming because the camphor is oriented very nicely on top of the iron where the ferryl oxo species is formed. So, in the next class we will look at the dioxygenase and some example, or even monooxygenase, which is not aromatic substrate or any kind of these ones. Then look at some other methane activation also we can see. Thank you very much.